It is my absolute pleasure to have the opportunity for presenting at the honorable stage of ISMB. Introducing myself, my name is Zeeshan, and I'm one of the faculty members at the Institute for Health and Robert Wood Johnson Medical School at Rutgers. I'm also the presenting author of our accepted and invited brief presentation entitled Human Gene and Disease Associations for Clinical Genomics and Precision Medicine Research to Investigate COVID-19. Precision medicine is one of the most recent and powerful developments in medical care. Further elaborating, it has the potential to improve the traditional symptom-driven practice of medicine with integrative and intelligent analysis of patients' personalized details involving lifestyle, clinical, genomic, phenotypic, and metabolomic profiles. Personal clinical profiles mainly include information about patients' visit to the practices, conducted diagnosis, lab tests, medications, and if there are any procedures. Question arises, how can precision medicine improve personalized treatment with predictive diagnostics? A simple answer to this is that one medication might be effective for one patient and might not be for another with the similar symptoms. To investigate it further, we need to look into the genomic profiles, which includes exploring information encapsulated within gene, transcripts, and mutations. We need to perform integrative clinical genomic data analysis, and for that we need comprehensive gene disease annotation repositories. It's been over 100 years since the word gene is around and progressively evolving in several different directions. Time to time, technological advancements have heavily revolutionized the field of genomics, especially when it's about, for example, triple code development, genetic mapping, data banks, gene disease maps, big data, next generation sequencing, catalogs of human genes and disorders. Variations in genes may lead to one or multiple causes of known and life-threatening, infectious, chronic, acute, and complex diseases, for example, cancer, diabetes, and COVID-19. Therefore, it is very important to understand the relationships among identified genes, mutations, and human diseases. To efficiently fathom from the wealth of genomics and clinical data, there is a crucial need to develop high-volume, clinically significant databases, bioinformatic tools, and machine learning approaches for gene disease annotation and integrative analysis. However, many such databases and tools are available worldwide. Recently, we have published a very detailed study and have reported comparative analysis of multiple available gene disease databases among different other findings, including their effectiveness and support for clinical genomics and precision medicine research. One of the most important issues around is that existing gene and mutation databases are nascent and incomplete in accommodating standardized clinical needs for connecting genomic variants to phenotypes and improving actionability and utility of genomes, expertise for the description and interpretation for structural and non-coding affecting variants. Defining the scope of our study and to support our precision medicine research, we started building a gene disease database, which so far have been concluded with over 90,000 variable gene disease associations, accessible through a smartphone app, and have been reported in recent publication. Next, we integrated information linking disease and drug codes into our database. Disease codes include the list of International Classification of Disease 9 and 10 maintained by the World Health Organization. When treatment codes include the list of over 100,000 medications organized by the Food and Drug Administration in the United States, also known as NDC, National Drug Codes. Extending further the scale of our database, we curated and added datasets including information about germline and somatic mutations. Germline mutation dataset includes over 100,000 classified SNP disease combinations based on over 67,000 SNPs reported for over 19,000 genes. Whereas somatic mutation dataset covers human genes with over 1 million SNPs coding mutations and their associated diseases. We have made this cutting-edge gene SNP disease drug database accessible through the latest version of our app. It contains consultation to several publicly available gene disease databases. Since last few months, an infectious disease, coronavirus, has affected the world. Despite many significant scientific and medical discoveries, the genetics of COVID-19 remains far from clear. Highlighting the usefulness of our app and database to this pandemic situation, users can look for the COVID-19 relevant disease-specific information, which includes clinical details, genes, and variants. Our most recent publication is available with further details about our database and app and their effectiveness to the current and future clinical genomic and precision medicine research. Concluding this presentation, I would like to appreciate the contributions of co-authors, Dr. Saman Zeeshan and Dr. Dong. I acknowledge the direct and collaborative efforts and pay special thanks to the current and former students of my lab. In the end, I would like to appreciate and thank for your time in attending this presentation. Should you have any questions, please feel free to get in touch at given contacts. Thank you very much.